ASRB net plant pathology. We have prepared a couple of model question papers, the preparatory model question papers, MCQ based, fill in the blanks, match the following, all kinds of various kinds of preparatory model question papers, not only meant for net but also meant for various state entrance exams especially related to plant pathology. My name is N.S. Shankar Reddy, PhD plant pathology. So we just made some, some previous questions and combination of various questions that will be useful for entrance exams. So we have prepared a couple of model questions. So let's go one by one. So the first one is the world's first plant production convention was held in London in the year. They're asking year. So the first model world plant production convention was held in 1929. So the next one is the first International Congress of Plant Pathology was held in London in the year. So the International Congress of Plant Pathology was started actually in the year 1968. So when it comes to what is International Congress of Plant Pathology, so where a famous plant pathologist all will comes together and they will discuss the problems and future prospects. So this happens once in every 5 years. It is actually started in 1968. So from 1968 onwards, for every 5 years, this International Congress on Plant Pathology was will happen in countries. So in 2023 it happens in France, Lyon. Of course, I have described. I have placed it in the next slide. So the second one, third one is the first Society of Plant Pathology and the oldest Society of Plant Pathology was, we have mentioned here, like the Netherlands Society, American Society, Indian Society, Phytopathology Society of Japan. So here the oldest Society of Plant Pathology is the Netherlands Society of Plant Pathology, which was started in 1891. So when it comes to American Phytopathological Society, it is the largest society of plant pathology that was started in 1908. When it comes to Indian Society of Plant Pathology, that was started by B.B. Munkur along with S.R. Bose in 1948. If I want to tell you the exact date, it was actually started in 1947, February 28. But it came to enforcement in 48. And then the last one is the Phytopathology Society of Japan, that was actually started in 1916. So they asked is the first society, and it is also the oldest society of plant pathology, that is Netherlands Society of Plant Pathology. So the next one is the Netherlands Society of Plant Pathology was established in the year, I think I have told already, 1891. So 1891, it is the oldest society of plant pathology and the first society of plant pathology that was started in Netherlands in 1891. APS stands for American Phytopathological Society. As I told you already, American Phytopathological Society is one of the largest society of plant pathology, which was started in 1908. So this American Phytopathological Society will publish a couple of books. I think you plant pathologists might have seen the various kinds of the good quality books that was published in APS uh, APS publishers. So APS actually stands for American Society of American Plant Pathological Society, which was comes under American Society of Plant Pathology, American Plant Pathology Society, which was started in 1908. So as I told you, International Congress of Plant Pathology, right? It is actually a non-profitable organization, especially promotes the plant health and as well as the security worldwide. As I told you that every 5 years, the International Congress of Plant Pathology will held. And if you see the first Congress, International Congress was happened in London, UK. The second one is exactly after 5 years, 1973, Minneapolis, USA, and 78 Munich, Germany, and the fourth one, fifth one, sixth one, and the last one that happens is Lyon, France in 2023. The meeting was already finished. I think it happens in August. And the next one, it is going to happen in 2028. I think it's going to happen in maybe Queensland, Australia. I'm not sure. Maybe Queensland, somewhere else in Australia. So the next one is we talked about Plant Pathological Society, right? Like the oldest society or the first society of plant pathology, that is the Netherlands Society of Plant Pathology. So which was started in the year 1891, and it publishes a journal called Netherlands Journal of Plant Pathology that started in 1895. It is a monthly journal. And when it comes to the next one is a British Mycological Society, then American Phytopathological Society. We just now spoke about American Phytopathological Society, right? That was started in 1908. The next one is Phytopathological Society of Japan, 1916. On the right side, I have mentioned the what are the journal that was published by. And the next one is Canadian Journal 1929, and the Chinese Society of Plant Pathology also started in 1929. But when it comes to India, Indian Phytopathology Society was started in 1947. So it publishes a journal called Indian Phytopathology. So in 1948 it was founded by B.B. Munkur along with S.R. Bose. And then annual reviews and various couple of pathological society I have mentioned here. But the first one is the Netherlands Society of Plant Pathology, and the Indian Society of the Indian Phytopathology Society, that is otherwise called as IPS, Indian Plant Pathological Society. American Phytopathological Society was actually established. I think we have already spoke about this American Phytopathological Society, which was actually established in the year 1908. And the next one is the Phytopathological Society or Phytopathological Society of Japan, that was established in the year 1917. And the next one is that is the Indian Phytopathology Society, which was established in 1947, February 28, if I want to tell you the exact date. And if the option of 1947 was not there, you can go with the option of 48, because it came into enforcement or complete action at 1948. That was founded by B.B. Munkur. And the next one is the Indian Society of Mycology and Plant Pathology, was started at the year 1971. So these are all the societies and their establishment dates. We may expect, I think in the previous year net question paper, I think 2023, they have asked what is the oldest society of plant pathology or the first society of plant pathology. I think we already spoke about. That is the Netherlands Society of Plant Pathology, that was established in 1891. And the largest plant pathological society in the world, I think I already told you, American Phytopathological Society is the largest society of plant pathology. And the next one is the first phytopathological periodical journal of mycology. I repeat, the first phytopathological periodical journal of mycology, which was initiated in the year 1885. Like at the time they used to publish maybe new discoveries or maybe some kind of new findings or some kind of new initiatives, whatever that was there, that he used to publish in this first periodical journal of mycology, that was actually initiated in around 1885. The next one is the annual review of phytopathology, was published in the annual review of phytopathology was started in the year 1963, and the journal of phytopathology is published by, I think every society they used to publish, or of course society means many scientists also be there as a part, and all the new contributions and discoveries, if they want to publish, they need to have at least some kind of journals to publish their discoveries, right? So every journal have their own discoveries, I mean sorry, their own society has their own journals, so that they will publish the papers. So here the Journal of Phytopathology was actually, that is related to American Phytopathological Society. If you see the previous slide or the second slide or maybe third slide, I have mentioned the journal here along with the what is the journal is related to which society or which phytopathological society that I have mentioned. So the next one is the Regional Journal of Phytopathology Mediterranean is published by, again it is same, American Phytopathological Association. So the Journal of Phytopathology and the Region Journal of Phytopathology in Mediterranean was published by both American Phytopathological Society or Association. And the next is the title of Journal Physiological Plant Pathology was changed to Physiological and Molecular Plant Pathology in the year. I repeat, I think this question was asked in somewhere, not in net exam, maybe in I think it's years before I saw it. I saw this question in somewhere else in college entrance exams. So initially the Journal of Physiological Plant Pathology, later name changed to Physiological and Molecular Plant Pathology in the year 1986. So the Journal of Mycology and Plant Pathology is published from, so it is a very important one. The Journal of Mycology and Plant Pathology was actually published from RCA Udaipur. Okay, it is a very important journal and very famous journal in India. So the Journal of Indian Journal, sorry, the Indian Journal of Mycology and Plant Pathology is presently known as, so the before Indian Journal of Mycology and Plant Pathology, the name was actually replaced or changed into the Journal of Mycology and Plant Pathology from 1997. So from 1997, Journal of Mycology and Plant Pathology was known as Indian Journal of Mycology and Plant Pathology was known as Journal of Mycology and Plant Pathology. That name changed to 1997. So Indian Phytopathology is actually published from New Delhi. There is a journal that is called Indian Phytopathology. So this journal is also very very important. For example, in ASRB, in maybe in the main entrance exams or maybe in interviews, they may ask such kind of questions. So there is no other choices, you must learn. And the next one is the first director of Empirical Bureau of Mycology of International Mycological Institute, Q. Actually this Empirical Bureau of Mycology actually located at Q, England. It was established in around 1920, I think. I'm not sure exactly, so most probably it's 1920. So the first director is E.J. Butler. So E.J. Butler, we know very well, he is considered as father of plant pathology and mycology in India. He is the first director of Empirical Bureau of Mycology, that was actually there in Kew, London. I think it was started in 1920. Just you guys can refer. The first president of Indian Journal of Mycology and Plant Pathology. The first president of Indian Journal of Mycology and Plant Pathology is R. Prasada. So this Indian Journal of Mycology and Plant Pathology was actually started in 1970. So the first president of this Indian Journal of Mycology and Plant Pathology is R. Prasada. The next one is who introduced the first course of plant pathology at Harvard University. So the first course of plant pathology in Harvard University was established by M.A. Farlow at 1875. The first course they introduced is diseases of crops. So whatever they identifies or whatever they found the new things related to diseases of crops, so they used to teach in 1875. He is the first one who introduced this plant pathology as a university course. He is the only pioneer at the time, and he is only used to taught diseases of crops. I think I have read his biographical memories. So in that he have mentioned he is the only one who started the teaching of disease of crops and for the first time in 1875, and later other scientists also came up with him, like they started like bacteriology and mycology. So such kind of things later came in 1910s and 1920s, but the first course of plant pathology in Harvard University was introduced by M.A. Farlow in 1875. So the next one is the famous book Advances in Fungi. So the Advances in Fungi was written by E.C. Lodge. It is a very famous book. I think this book was published in 1953. I'm not sure. Probably 1953. So the next one is the textbook Plant Pathology was written by. It's a very famous book. The textbook of Plant Pathology was written by George N. Agrios. There are 5 editions. Now the 6th edition also came in 2023 August. The 6th edition is also published. So far there are 6 editions of this plant pathology book was published by G.N. Agrios. It is generally considered as Bible of plant pathology. It's a very famous book that can be used for PG, PhDs and various teaching staff also. It's a very famous book that is generally considered Bible of plant pathology, so which was written by G.N. Agrios. There are 6 editions are there. The 5th edition was published in 2005 and the 6th edition was published in 2023 around August time, which was edited by Oliver. Okay, so previous 5 editions was written by George N. Agrios. The 6th edition was edited by Oliver because the Agrios was no more now. The second one is Hunter Sitchin Ober was authored by, I repeat, Hunter Sitchin Ober was authored by Anton de Barry. It is the first work that was made by Anton de Barry. So even though he is a doctor, originally he is not a plant pathologist, he is not a mycologist, he is not a botanist. He is originally a doctor, but even though he is a doctor, his thesis is MBBS thesis is generally related to botany or generally related to plant pathology. His first work was Anton Sitchin Ober. It is actually German language, and later we know that Anton de Barry become the great pioneer in plant pathology, and we generally considered as father of plant pathology. In fact, I have published one newsletter regarding Anton de Barry, where I have mentioned from top to bottom on the hundreds of students that he have trained, everything that I mentioned in the article. That will be just dwell into the history again. I hope that will be useful. If you want, you can just go and check again. And the second one is Matthew's Plant Virology was written by Roger Hull. We know very well. So for generally plant pathology, Anton DeBerry book was very famous. When it comes to plant virology, Roger Hull. So Matthews plant virology, Roger Hull plant virology, we used to see in the libraries, right? So this Matthews plant virology was written by Roger Hull. The next one is a renowned scientist Norman E. Borlaug, belonging to the field. Actually, we know very well this Norman E. Borlaug is generally considered as father of green revolution. He is actually a plant pathologist and he is also a agronomist and a genetist as well. But since he is also a plant pathologist that I have mentioned in this question, he is not only a plant pathologist but also agronomist but also a genetist. The next one is in India, the first British man to get the disease specimens was identified by. So in 1870s and 1860s, we are under the colonial rule. So in India, the first British man who started identifying the specimens is Selman in 1931, Sleeman in 1939. So the later when it come to the identification of fungi in India was first made by. So in Sleeman he have done first collection of the specimens at the time. In later, the identification of fungi in India was first started by Cunningham and Barclay. So they both are in around 1880s. They started collection of identification of fungi. In India, when it comes to in India, the first Indian scientist who collected and identified fungi is K.R. Kritiker in 1885. Exactly 1885. It is a very important question and it was asked in many areas also. So the first Indian scientist who collected and identified fungi is K.R. Kritiker in 1885. The first empirical mycologist in India. So the first empirical mycologist in India is E.J. Butler. He is the first empirical mycologist, first plant pathologist and mycologist in India. He is actually not an Indian scientist. He is actually an Irish scientist. So since in 1880s and up to 1947 we are under the colonial rule. So he actually came as a botanist to India and later because of his discoveries and the greatest contributions he became the father of plant pathology and as well as mycology in India. So the first empirical mycologist in India was again E.J. Butler only. So the next one is monograph of Pitheaceous and allied fungi was authored by. So the monograph of Pitheaceous and allied fungi was again authored by E.J. Butler. So this is a little bit history related, right? If you want to know much more about the history, we have published lots of videos regarding history of plant pathology, history of mycology. All these videos are we have already published. If you go there, you will find much more information than what we are discussing here. So generally when it comes to this question, monograph of Pitheaceous and allied fungi was written by E.J. Butler. And of course he wrote many books and diseases of oh sorry, not diseases, and he wrote very famous book uh, fungi and sorry, actually I forgot. I think fungi and diseases in plants. I think that is the very famous book that was written by him, Fungi and Diseases in Plants, 1918. That is a very famous book. That was actually the first foundation book related to plant pathology in India. So E.J. Butler, his book is the Fungi and Diseases in Plants. The next one is who is a father of plant pathology in India. Just now we discussed E.J. Butler is generally considered as father of plant pathology in India. The next one is fungi and diseases in plants. Okay, now it came right. So the fungi and diseases in plant was written by E.J. Butler, Irwin John Butler in 1918. And there is one more book called Fungi and Plant Diseases that was written by B.B. Munkur. Fungi and Diseases in Plants. Fungi and Diseases in Plants that was written by E.J. Butler. Whereas Fungi and Plant Disease was written by B.B. Munkur. So this Fungi and Disease in Plant was published by E.J. Butler in 1918. The next one is who is the father of plant pathology in India? I think just now we had discussed E.J. Butler. And again the same question, the fungi and disease in plants, which was written by E.J. Butler only. The next one, the green ear or downy mildew of pale millet was first time reported in India by E.J. Butler. So why he become the father or the greatest contributor? Because of various kinds of discoveries and identifications. If you see the important diseases in India, the major breakthrough discoveries in India, mostly it came from the diary of E.J. Butler only. So the green ear or downy mildew of pale millet was first time reported in India by E.J. Butler only. So the next one, a new disease of castor oil plant that was discovered in 1913 by. So in India, the first time JF Daster, he discovered or established a new genera called Phytophthora genera in India. So he's an internationally renowned for the establishment of Phytophthora genera in castor. So he's the first Indian scientist who discovered that a new disease in castor oil plants, uh, that is JF Daster, around 1970s and 1980s, in between time, and it is a very famous disease. Vivotoxin. So the concept of vivotoxin was actually given by T.S. Sadasivan. So the term vivotoxin was coined by Diamond and Wagner in 1953. That term, but the concept was given by T.S. Sadasivan. It's a very important one. So the next one is the seedling blight. Just now we told in India, the castor, that was a new disease, or Phytophthora genera, was first time established by J.F. Daster. The disease name is seedling blight of castor. The seedling blight of castor, which is caused by Phytophthora parasiticae, were first time recorded in India by J.F. Daster. J.F. Daster was internationally renowned for the establishment of Phytophthora genera in castor. The disease is seedling blight of castor. In exams, we can also expect. So, what is the disease that was discovered by J.F. Daster in castor? That is a seedling blight of castor, which is caused by Phytophthora parasiticae. And the next one is L.C. Coleman, is generally a plant pathologist during 1914, and he worked and suggested for the control of which disease? Lead blight of potato, tear wilt, blister blight of cocoa, and coliroga of areca nut. So here this L.C. Coleman actually worked in South India. At the time he recommended the management practices for coliroga disease of areca nut. This coliroga disease of various name, bud rot, or other various kinds of diseases are there. Mahale disease, tanjore wilt, various kinds of other names are also there, right? So the next one is solar energy treatment or solar heat treatment for the management of wheat loose mud was actually suggested by Luthra and Sutter, I think in 1953 time. So solar heat treatment, in the sense by using or by trapping the solar energy, which means sunlight only, nothing else. By trapping the sunlight, the management of wheat loose mud was actually suggested by Luthra and Sutter. So this is very simple principle, and most important thing we need to remember is this loose mud management, especially by using the solar heat treatment, is especially recommended for summer periods only, where the summer temperature is high. So if the summer temperature is low, this treatment is ineffective. Very simple. Seeds are treated, seeds are soaked in water and then sun dried. Then before planting, we can use seed treatment fungicide like Thiram or Captain, whatever it is. Very simple. Seeds are soaked in water for 4 hours, maybe 8 to 12, and then again sun dried for 4 hours, 12 to 4, in a cement floor, so that the temperature is high. Automatically, if there is any fungi will be, or maybe mycelium will be there inside, will be die. So that is the principle, the solar heat treatment. As I told, it is especially recommended during summer periods. Used for loose mud of wheat. Slagonoda triticum. The next one is extensive studies on mango black tip disease was carried out by. So S.N. Dasgupta, he is a very well known and renowned for the especially in the study of black tip of mango. So this is caused by two reasons. One is nutritional disorders, and then second one is normally pathogens. Generally, this sulfur related toxicity, if the bricklings are nearby this mango fields, due to this sulfur toxicity also, there is this disease will come. Black tip of mango, which means at the tip of mango we can see black color dots, that is called black tip. Sometimes we may confuse with anthracnose also. This black tip is generally maybe boron deficiency or sometimes sulfur toxicity also. The next one is the disease of croplands in India was written by, it's a very famous book that was written by G. Rangaswamy, which was published in PHI Learning Publishers. Disease of croplands in India. Actually, it was written by two members, G. Rangaswamy and R. Mahadevan, I think so. And the next one is bacterial plant diseases in India was written by. In India, the bacterial plant disease was written by again same G. Rangaswamy only. Phytopathometry was written by. So the phytopathometry was actually I think it's related to C.D. May. I think so. The term phytopathometry was also the term phytopathometry was actually introduced by E.C. Large, I think, but this book of phytopathometry was written by C.D. May, that is related to how to measure plant diseases like disease intensities or all kinds of things that was mentioned in this phytopathometry. The second one, the next one is kernel bunt of wheat was reported in India for the first time in 1931 by Mitra. So here why it is called kernel bunt, because the disease was first reported in the agriculture station of Kernal. So Kernal is an area, hope we guys know very well. So Kernal is an area. In Kernal there is an agriculture research station. In 1931, Mitra was first to report this disease, that is a kernel bunt of wheat in 1931. It's a very important disease. Kernel bunt of wheat in India was first reported by Mitra in 1931. And one more question was there, this kernel bunt infected fields, actually Neovacia indica, before it's called Tilicia indica. We can also call it as a Neovacia indica, okay, but the Neovacia genera was created by B.B. Munkur. So initially it was called as Tilicia indica by Mitra, and later the spores, that is sporidia which is produced, that is more related to Neovacia, so that the B.B. Munker placed into Neovacia. Again other societies came into existence. No, by using only sporidial character we cannot come to a conclusion that is belong to Neovacia. So again the name is restored into Tilicia indica. And if you see the literature, both names will appear, Tilicia indica, Neovacia indica. But the Tilicia indica was given by Mitra only 1931. But Neovacia indica, the genus Neovacia was given by B.B. Munkur. This kernel bunt of wheat infected peels, that generally fishy kind of smells that we can observe. The reason is it produces a chemical called trimethylamine. The infected kernels are there, right? The infected kernels are completely becomes smut balls or smut spore balls. That smut spore balls generally exhibits a fishy smell due to the presence of a chemical called trimethylamine. It is actually alkaloid, I think. So trimethylamine. The next one is the book Fungicides in Plant Disease Control was written by. So it's a very important book. It's a very famous book also. The Fungicides in Plant Disease Control was written by Y.L. Neen and P.L. Tapilial. The first edition was I think published in 1971. So I think now fourth edition is running up. The Fungicides in Plant Disease Control was written by Y.L. Neen and P.N. Tapilial, where they have mentioned only fungicides, advanced generation fungicide, what are all the various kinds of fungicides were there from since the starting to now, and along with the various groups of fungicides like heterocyclic nitrogen compounds, various kinds of classifications of fungicides are there, right? That was the very clear and neatly described. But that was written by Y.L. Neen and P.N. Tapliel in 1971, I think. The next one is the textbook of plant diseases was written by the textbook of plant disease was written by R.S. Singh. Is a very famous plant pathologist. Is also considered as a lighthouse of plant pathology because he guided many students. Now they are in the field of ARS. So he is such an outstanding plant pathologist, and I think he died in 2018 around. I think, I'm not sure, maybe 2018 or 2019. He wrote so many books. We know very well that plant diseases or disease of crops, so many kinds of books was written by him. But the textbook of plant diseases, that was an outstanding book that was actually written by R.S. Singh. The next one is the word plant pathology is derived from the word at the term plant pathology is derived from Greek word pathos and logos. Hope you guys know very well pathos and logos, just study of plant pathogens for your understanding, very simple. So the next one is in 1942 and 43, Bengal faced very serious famine, that is called Bengal famine, that was due to Helminthosporium varese, also called as bipolaris varese. So it is a pathogen we know very well, brown leaf spot of rice, that is actually the responsible for Bengal famine. So there are two famines which are very famous famines when it comes to plant pathology. Irish famine that happens in Ireland, that is late blight of potato Paedobdera infestans. The second one is a Bengal famine, that is a brown leaf spot of rice which was caused by Helminthosporium varese, can also called as bipolaris varese. If you see the teleomorphic stage, Cochleobolus mia benus, I think. So the next one is in India, leaf rust of coffee was first time recorded in around in 1870. The leaf rust of coffee was first introduced and first identified in India. It is actually introduced from Sri Lanka because at the time coffee production and tea production was much. Actually, in 1870s and 1880s time, Sri Lanka is generally considered as coffee hub to the entire world. Around 90% of the coffee that was exported from the other countries that was originated from Sri Lanka only. The Britishers used the Sri Lanka as a coffee hub. So in around 1860s and 1870s, the Sri Lankan coffee was infected by a disease called coffee rust, Hemilia vastrastris, that wiped out almost 90% of the coffee population. But when it comes to India, the leaf rust of coffee was in India was first time recorded in 1870 around. The next one is the wilt disease of sugarcane was first time reported in Bihar. The wilt disease of sugarcane was first time reported. The next one is the information of the spread of black, brown, and yellow rust through field surveys in India was first identified by L.M. Joshi. The next one, cause and control of Kaira disease of paddy or Kaira disease of rice was first investigated by Y.L. Nine. This Kaira disease was actually first reported by Y.L. Nine and P.N. Thapilial around 1970s and 18s time, in between I think late 70s I think. So this Kaira disease is actually a nutritional disorder. This is also very important disease which is actually due to zinc deficiency. If the fields are low in zinc, this disease we can see just like yellow color discoloration, especially on the tips can be seen. That is the identification feature. So this Kaira disease was first time reported by Y.L. Nine around 1971 or maybe late 1970s I think, I'm not sure. So Y.L. Neen and P.N. Thapilian were the first to discover this Kaira disease which was caused by zinc deficiency. Then how can we manage this? This is very simple, application of zinc sulfate that rectifies this Kaira disease, because it's a nutritional disorder. We can easily rectify if we supply the proper amount of nutrients. That's it. The next one is the citrus ring spot disease was first time reported from India. In India, the citrus ring spot was first reported by Ahalwat in 1989. The next one is most commonly used surface disinfectant. Here two kinds of surface disinfectant generally used, sodium hypochlorite and as well as mercury chloride. But mercury chloride was banned. Now we are using sodium hypochlorite in the laboratory. So if you see, the concentration is also very important, 1%. Sodium hypochlorite 1%. If it is a mercury chloride, 0.1% can be recommended. 0.1, because mercury is very toxic, right? So majority of the laboratories are using sodium hypochlorite only, but of course mercury chloride is also available. If it is a mercury chloride, the recommended concentration is 0.1. If it is a sodium chloride, the recommended concentration is 1%. The next one, fifth edition of plant pathology, that was GN Agrius, I think we have already discussed, which was published in the year 2005. The first edition was published in 1969. So the later editions like second edition 78, third edition 87, fourth edition 1997, the fifth edition is 2005, and the sixth edition is 2023 August around. So if you see the pore size of laminar airflow chamber, what is laminar airflow chamber? Generally it is considered as the heart of the laboratory. You guys know very well. So whatever the experiment we used to perform, we'll do inside the laminar airflow chamber only. So inside it contains a filter called HEPA filter, high efficiency particulate air. So the general size or pore size of this HEPA filter is around 0.3 micrometer. It's a very important, 0.3 or 0.33 micrometer. The next one is Central Potato Research Station is located at. So here we guys know very well the potato can be cultivated in Ooty, Darjeeling and Shimla, but the Central Potato or CPR is located at Shimla. Okay, the next one is if I want to tell you exactly where it is, Kufri village of Shimla. So the next one is, that's why the majority of the varieties whatever we are publishing, Kufri Jyoti, Kufri Navin, all these potato varieties are starts with Kufri. Kufri is the village name or the place name where the research institute is located at. And the next one is the Indian Sugarcane Research Institute is located at. It is located at Lucknow. Sugarcane Breeding Institute is located at Coimbatore. Okay, there is a fundamental difference. I repeat, Sugarcane Research Institute is located at Lucknow, Uttar Pradesh. Sugarcane Breeding Institute is located at Coimbatore, TNA, Tamil Nadu Agriculture University. We know very well, right? So that is actually this breeding institute are in collaborated with this TNA Coimbatore. The next one is the term hydrochory and anemochory was actually coined by government. Hydrochory means the spread through water, anemochoria through spread through air, that is. So the next one is the enzyme used for binding of primers. Actually, uh, so the enzyme which is used to bind is DNA ligase. So restriction endonuclease is the enzyme which is used to cut the DNA, or which is used to cut the genetic material. The cutting enzyme is called restriction endonuclease, whereas the binding enzyme is called DNA ligase. Ligase means it binds. Okay. The next one, same. Enzyme used for cutting, restriction endonuclease. Just now we spoke. The enzyme used for binding is DNA ligase. Enzyme used for cutting is restriction endonuclease. The next one is 10th International Congress of Plant Pathology was held in. So the 10th International Congress of Plant Pathology was held in China. Before we saw the International Congress of Plant Pathology, the first one is the London 1968. For every 5 years, once this International Congress of Plant Pathology happens. In the next, or I think 14th or 13th or maybe 14th, the International Congress of Plant Pathology is going to held in Australia, Queensland, I think. So the 2023 already happened in Nathan Lyon, France. So if you want to go for this international congress, you must I think record now itself. Maybe it will cost around 5 to 10 lakhs, because for going, coming, everything all puts together. But it's definitely worth of going, because it's a once in a lifetime moment for us, because the next one, the term taxonomy was coined by. The term taxonomy was coined by D. Condole. The term taxonomy was coined by D. Condole, but the father of taxonomy is Linnaeus, because he is the one first introduced binomial nomenclature and also classified organisms like into plant and animalia, like that. Such classifications was given by him, but the term taxonomy was given by A.P. D. Condole. The next one is the active ingredient present in the eucalyptol. These plant products can be used in the management of plant diseases, right? So vector used in human genome project. In human genome project, we know very well human genome project, it is started around 1990 and finished in 2003. Almost it took 13 years. So at the time they used a vector called yeast artificial chromosome, that is the vector used in human genome project, HGH. And the next one is which of the following is a neutral stain? They're asking neutral stain. Neutral red is very simple, the answer is there in here itself. And the next one is the method in which the cells of frozen dehydrated. So generally frozen cells or frozen condition of maintaining the cells is called lyophilization. The next one is who demonstrated the technique used to avoid all microorganisms is generally accomplished by sterilization. So this sterilization is a technique where we can kill all the microorganisms for a microorganisms free environment, so that no contamination will occur. Okay, so the next one is the first antibody to contact invading microorganisms. There are 5 kinds of antibodies are there if you see in virology. So soon we are also planning to do videos on plant virology. I think we have already published plant virology, but still we are planning to do more advanced plant virology related videos. So the first and foremost antibody that contacting the invading microorganisms is IgM. So this is the first antibody that used to connect with the microorganisms. In a fluorescence microscope, the objective lens is generally made up of. So in a fluorescence microscope, the objective is generally made up of, I think, polythene. I will check it once again. You guys also please check it once again. So I think it's a polythene. So the next one is which of the following fungi is the most serious threat in a bone marrow transplant unit? So Aspergillus. Generally, Aspergillus fungi is a very serious threat when it comes to bone marrow transplanting unit. It is not only this bone marrow transplanting unit. If you see all the cultures in the laboratory is also generally contaminated by this Aspergillus, Penicillium, and Rhizopus. Okay, algae is rich in. Algae is generally rich in all kinds of stuff like proteins, carbohydrates, or vitamins. We can see single cell proteins. Single cell proteins are rich in vitamins, proteins, and as well as carbohydrates. This is actually generally algae. In space research also, scientists are now planning this growing of algae, because it contains rich amount of carbohydrate, vitamins, and as well as proteins. So there is no need of carrying of large stuffs and containers, especially for food. So if you grow algae, it will be very easy to to supply all kinds of nutrients to the human body, include proteins and vitamins and as well as carbohydrates. So generally algae is generally rich in all these three kinds of substances. The next one is, so the image obtained from a compound microscope, generally the image obtained from a compound microscope is a virtual image, okay? It's not a real image. In compound microscope we can only see a virtual image, okay? So the next one is infrared radiation is a method of sterilization by, so generally this infrared radiation is I'm not sure, probably this infrared method of sterilization, probably mechanical method. Just go through once again, please. So the next one is the antiseptic method was the first demonstrated by Lister. So the Lister was the first one who demonstrated this antiseptic method. So the next one is lyophilization means freeze drying. So just the rapid freezing of a microorganism so that we can maintain the culture for a long-term preservation. So long-term preservation generally done at a liquid nitrogen stage or in a freeze drying stage. So that is called lyophilization, where we can rapidly dry the product or rapidly cool and dry the product in a equipment called lyophilizer. So where we can convert into powder formulation. If you see milk we are converting into powder, right? So like that we can also convert this cultures into the powder formulations, so that we can preserve for long-term preservation like 5 years or 10 years, 15, 20 years, also up to 20 years we can maintain, I think. The next one is smallpox vaccine was the first developed by, so the first vaccine was actually developed by Edward Jenner, that is a smallpox vaccine. So the next one is which agar plate method is commonly used for the estimation of the number of bacteria in a milk. Generally standard plate count is used to estimate the number of bacteria. The next one is bacterial spores can be killed by 70% alcohol, glutaraldehyde and autoclaving. So both B and C can be recommended, that is glutaraldehyde and autoclaving can be recommended for bacterial spores, because the bacteria, if you see some this endospores, so generally bacteria don't produce any spores, okay? Fungi only produce spores, but bacterial spores are generally called as endospores. These endospores are highly heat resistant, that can survive high temperatures. So the only technique we can kill this is there is a technique called tyndallization. So generally like chemicals like glutaraldehyde and autoclaving generally used for killing of this bacterial pores, since they are highly resistant. So the next one is in electron microscope, which material is used as a objective lens. Magnetic coils are generally used in electron microscope as a objective lenses. So for example, if you see compound microscope, we can see the object. If you want, you can also remove the objective lenses if you want, right? So but in electron microscope, magnetic coils are generally used as an objective lenses. In electron microscope, there are two types of electron microscopes are there: SEM and TEM. In general I'm telling, okay? SEM means scanning electron microscope, TEM means transmission electron microscope, which costs around 60 lakhs to 7-8 crores up to. So this the first time this electron microscope was invented by Knoll and Ruska in 1932. So Knoll and Ruska are the first scientists who invented the microscope, that is electron microscope, in 1932. So the later the prototype was developed by Ruska. He also got Nobel Prize in around 1980s and 1982, I think, just because of this mechanism of developing this microscope and other stuff. The next one is glasswares are sterilized by, so glasswares can be sterilized by autoclaving and hot air oven also, but since they have given in a way that like where we are generally using sterilizing, so hot air oven, because this autoclaving is for killing, right? So autoclaving is generally for killing. So the temperature used to maintain the temperature generally used to maintain in the autoclaving is 121.6 degrees Celsius for 15 minutes at 15 psi pressure. This Charles Chamberlain was first to introduce this autoclave. This is used for killing, okay? But when you see hot air oven, after killing, if you want to sterilize, we can use another equipment called hot air oven. This hot air oven was actually introduced by Louis Pasteur. We know very well who is Louis Pasteur, right? So this pasteurization technique for milk and other kinds of beverages at 62.8, though the temperature may varies to beverage, like if it is a milk 62.8, generally for other beverages it may varies. So generally this is the hot air oven. and if the temperature increases, the amount of time to sterilize an hot air oven goes down. So there are some temperatures 120 degrees, then you can go for 6 to 8 hours, 180 then 30 minutes, or 190 12 minutes, something like that. If the temperature goes up, the time period for sterilization goes down. Generally we can recommend 160 degrees Celsius for 1 to 2 hours. That is the standard, some general standard. But if you want to work a little bit faster, then you can go for 180 up to 190 degrees Celsius. So the next one is the best method for getting a pure culture is, so pure culture generally we can for example, if you are isolating from any culture, though, so this normally contamination will occur. So there are a couple of methods which are used to getting the pure cultures, like streak plate method, pour plate method, or some spread plate method. Some kind of techniques are there. So here, when it comes to here, both this streak plate and agar slants are used for getting the pure culture, because if you see agar slant, the space will be very less and the contamination chances also very less. And moreover, if you see in petri plate, the storage time is much more higher when it comes to agar slant comparatively in petri plate, because the diameter of petri plate is more, so the availability space is also more, so that the fungi or the other kinds of microorganisms will go faster and utilize the nutrient faster. So when it comes to agar slant, the amount of the area is less, so that we can store the microorganisms for a long period, as well as very little contamination. No contamination almost will happen when it comes to agar slant. It's my personal experience. And the next one is the resolution power of a compound microscope. Generally the compound microscope can resolute up to 0.2 microns. And the next one is autoclave. The principle of autoclave is moist heat and as well as steam under pressure. So generally, if I want to tell you only one, steam under pressure, like how we are preparing idli, this is the same principle, autoclave also, because it contains water. When we are using an electrical current, so the water will boil up. The water also just generally convert into steam under pressure, so that the microorganism will die, and also moist heat, right? So both B and C. The next one is the capacity of a given strain of microbial species to produce a disease is generally known as, what they are asking is the ability of a pathogen to cause the disease is called as, generally it is called as virulence. Virulence in the sense, ability of a pathogen to cause the disease is known as virulence. The next one is acridine dye is generally most effective against, acridine dye is generally most effective against the gram positive bacteria. So then in bacteria there are two kinds of bacteria are there. Hope you guys know, gram positive and gram negative. Generally these acridine dyes are more effective against this gram positive bacteria. So in majority of the plant pathogenic bacteria comes under gram negative, but there are very few bacteria that comes under gram positive. For example, since all our plant pathology started our life with BSc agriculture, right? BSc. B stands for Bacillus, S stands for Streptomyces, C stands for Corynebacterium or Cortobacterium, other kinds of bacteria. These are all the three important bacteria that comes under gram positive: Bacillus, Streptomyces, Corynebacterium or Clavibacter also. These are all gram positive bacteria, but the remaining majority of the plant pathogenic bacteria are gram negative, like Xanthomonas, Pseudomonas, all those things are gram negative. The next one is hanging drop method for motility study was first introduced by Leeuwenhoek. Antonio Leeuwenhoek was the first one who introduced this hanging drop method for motility. The next one is the sterilizing agent. Which of the following is a sterilizing agent? Ethyl oxide, oxygen, nitrogen, carbon tetrachloride. We know very well ethyl oxide, because oxygen and nitrogen is not less sterilizing agents, and carbon tetrachloride is also not. So this ethylene oxide is the sterilizing agent. The role of phagocytosis was discovered by, phagocytosis means eating their own cells. So that was first discovered by Metchnikoff. The next one is the principle involved in the streak plate method is, the principle generally involved in streak plate method is just a dilution. That's it, nothing else. Separation or streaking or isolation or dilution. Very simple, it's a dilution. So the next one is the ability of a microscope to distinguish two objects into separating objects is known as, generally it's a resolution power. So generally if you are separating the objects, or if you are seeing the objects, so this is generally resolution power. Two objects, like two separate objects, if you are seeing how you are resoluting. So that's actually depends on the answer is resolution power. The next one is alkaline alkalophiles grow at the pH. Of course, we know very well acidophiles, basophiles, neutrophiles, and alkophiles. So acidophiles generally grow at the temperature of 1 to 1.5, 1 to 5.5, around 6, 1 to 6. So generally base means or neutral means generally 7. So in between 6 to 8 or 9 is generally considered as 7. Alkali means above 7, in between 8, 9, something like that. It is called alkalophiles. So 7 to 12. The answer is 7 to 12. So who perfected a magnetic lens in 1927? So actually Geber, he was the first one who actually perfected a magnetic lens. A perfect magnetic lens was first developed by Geber in 1927. So the next one is an electron microscope. The source of electrons is from, generally the source of electrons is from tungsten metal. So the tungsten is a very costly metal. Hope you guys know it. In bulb also we can see this tungsten metal, right? So in electron microscope, where this source of electrons are coming from tungsten metal, because if you see in bulb also it contains the tungsten metals, right? So the tungsten is only emitting. The original tungsten can last on thousands of years. We know very well. The bulb was first invented by Thomas Alva Edison. When he discovered that first bulb, he used the original quality tungsten. That bulb is still lighting now. It's been almost 150 years since that discovery. Still that original tungsten bulb was lighting now. So the companies what we are using the tungsten bulb are their normal quality tungsten bulb. They have an automatic setup that after 2 or 3 years or maybe 4 or 5 years the tungsten automatically damaged, the bulb will go up again, because if they produced original or good quality bulb, so we are not going to buy new bulbs, because how they will survive? Because if it is a quality, how they can increase their market cap or market, right? So that's why the companies itself, they used to make normal quality bulbs, so that 4 to 5 years the bulb will goes off. So in electron microscope, the sources of electrons are generally tungsten. Okay, the next one, reduction of virulence is known as, normally we know the reducing the virulence or the due to continuous subculturing or due to continuous maybe way environmental conditions, the virulence may be reduced. So the reduction of virulence is known as attenuation. So attenuation is a phenomena where virulence of a pathogen can be reduced. Enhancement of a virulence, this is a new terminology, right? We only read about reduction of virulence, but there is another terminology is called enhancement of virulence, is known as exhalation. Exhalation is the terminology which is used for the enhancement of virulence. So the next one is the most important virulence factors are adhesions, invasives, toxigenicity, enzymes, and all the above. So when it comes to, depends on the pathogen, because when if you see, couple of pathogens produces enzymes as a pathogenicity factors, couple of pathogens produce uh, toxins or maybe polysaccharides or various kinds of growth regulators and hormones. So it vary with pathogen to pathogens. So which whatever I have mentioned here, adhesions, invasiveness, toxicity, and enzymes, all these are actually the important virulence factors. It may varies and depends on the pathogen, but the answer is all the above. So the next one, which of the following mineral elements play an important role in biological nitrogen fixation? Molybdenum plays, generally molybdenum plays a very important role in the fixation of atmospheric nitrogen. The next one is ability of a pathogen to spread in the most tissues after establishing the infection is known as, so invasiveness. I repeat, just now we spoke about what is in here we mentioned about invasiveness, right? So invasiveness is generally about the pathogen to spread host tissues after establishing the infection is known as invasiveness, invasion. For example, in recent war also, Russia is invading the Ukraine. Invading means it is spreading. It is spreading its geography. It's spreading its land. Invasion means so just spreading like infecting the further cell and spreading to all over the plant. It's simply invasion. Okay. The next one is the most important vitamin for the growth of bacteria. Generally, for the growth of bacteria, the most important required vitamin is generally B complex kind of vitamins are most important for bacteria. So most bacteria do not require cyan. Which ion is not required by the bacteria? So bacteria generally not requires sodium ions. So septicemia means, so septicemia, blood, bacteria in the blood, toxin in the blood, pus in the blood, multiplication of bacteria and toxins in the blood. So generally septicemia is a multiplication of bacteria in the blood, is generally known as septicemia. So the next one is beneficial phytoplasmal diseases. So little leaf of brinjal, sesame, phyllodes, poinsettia, phytoplasma infection and all the above. Generally phytoplasma, we know like it's a kind of plant pathogen which causes disease like fungi and bacteria. So here little leaf of brinjal and sesame phyllodes are the dangerous diseases, that is harmful diseases we know very well. And there is one phytoplasmal disease called poinsettia phytoplasmal infection. This is the beneficial phytoplasmal diseases which actually benefits the poinsettia plants. So then last one is tobacco and tea leaves are fermented to give flavors and the taste. So generally this type of fermentation is known as alcohol fermentation, curing, degradation, lactic fermentation. This is generally is called curing. So curing type of fermentation only, where this tobacco and tea leaves are generally gives a good taste and flavor. So we might have studied the curing only in tobacco, but curing can also be done for tea leaves also, that gives a very good taste and as well as flavor. So this actually the we what we have discussed is here is just MCQs related questions. Much more information was actually described in this book, A Vision into Plant Pathology Complete Student Version. This will be definitely helpful for masters, UG, PG and PhD levels, for not only NET but also for ARS or maybe various kinds of state entrance exams. If it is related to plant pathology, this will be definitely helpful, and it's, I would say, it's a pocket encyclopedia for books. You just buy it, you won't regret the decision. So further more information or further more guidance, you guys can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.